Uh, Good evening. So we will begin today's session. Fully I'm audible. So I will start sharing the screen. And so this session is actually was supposed to be taken last Sunday. However, I wasn't well, so I have postponed. This is the rescheduled week 6 session, as in tutorial session. So what we will do in this session is summarize week 6 lecture content and then look at few questions. Okay. So I will start sharing my screen. Okay. All right. So to begin with, in general, this is a molecular biology session where mostly we will look at the cellular processes, molecular biology processes that takes on in our body. Okay, so begin with, if we talk about the central dogma of life, okay, so what it basically looks at, what it tells is how the genetic information in our cells is actually encoding it in like red, and it encodes different kinds of other biological molecules, macromolecules, which in turn, drives all the life that we see around as well as drives all the metabolic reactions going on in our body. So if we talk about the central dogma, um, it is actually controlling the biological processes, and mainly there are three processes we can see in this pictorial representation as well. One is this replication, where it is uh, the genetic information on the DNA is actually again getting copied. The same thing is getting replicated. So this is why this is using different kinds of machineries, proteins, factors, etc. So this is replication. Next comes the information on the genetic information on the DNA is actually again read by certain factors, proteins, and other factors together, regulatory factors and enzymes. Then it encodes another messenger macromolecule, which is the RNA, and this process is called transcription. Okay. And then finally, the message that is encoded from the DNA and is carried by the RNA is actually again read by certain factories, which are protein synthesis factories called ribosomes to synthesize proteins. Okay. So mostly it is central dogma goes from like it mainly enlists three processes, which are replication, transcription, and translation. Now, one more thing is that it's usually earlier it was like always unidirectional, that always DNA will give rise to RNA, then RNA will be encoded to synthesize proteins. However, later when viruses came into picture, viruses were discovered and their replication machinery and stuff were discovered, then reverse transcription came into picture, where there are in general the genomic content is present in the form of DNA, but viruses, RNA is the genomic content in viruses, and what happens is RNA actually needs to be converted to DNA rather than the unidirectional flow of DNA to RNA. We have is RNA is present as a genomic content in organisms like viruses, and then it is actually reverse transcribed into DNA, and then the DNA further undergoes replication using the host's replication machinery, and this is again done by certain specific enzymes and regulatory factors. Basically, the enzyme that is the process is reverse transcription. Transcription is DNA to RNA is transcription. RNA to DNA is reverse transcription. Okay. And again, um, so reverse transcription, what is happening is it's actually reading the RNA template and polymerizing the synthesizing DNA, right? So RNA dependent DNA polymerase is being used in this for this purpose. Okay. Next, there's RNA replication as well. So in most of the organisms, genomic content is present in the form of DNA, right? We read about this. Although, but like RNA can also be duplicated via replication uh, from the sequence content or sequence information present on the parent RNA strand. Okay, and but in reverse transcription, what we saw is RNA is being used to synthesize DNA, and then that DNA is getting replicated, right? But and the purpose, that is, I mean, the enzyme that is primarily carrying out this process is RNA dependent. The template is RNA, and it is polymerizing a DNA strand. However, in case of RNA replication, it will be RNA dependent. Definitely, the enzyme is actually reading the message or reading the code on an RNA strand, but it is synthesizing RNA, so or polymerizing an RNA strand, so it is RNA polymerase. Okay. And sometimes DNA can directly encode a protein, but again, this can happen in a cell-free system inside the test tube, which is called in vitro cell-free system. So it is actually possible to translate DNA directly into the protein into protein sequence in the presence of ribosomes. Ribosomes are basically the protein synthesis factories. Okay. However, we cannot really precisely control it, and it is not really known whether it synthesizes the protein in a sequence-dependent manner. Okay, protein to protein. So there are certain infectious proteins which replicates to form identical copies of themselves, and these proteins are called prions. Although it represents the transfer of information, but this is not the usual pathway and considered to be the exception of central dogma. Okay. Now let's look at each of the molecular biology processes one by one. Okay, so there is DNA replication. So one is this replication machinery. So what really happens is one DNA strand, whatever is the, so DNA exists as a double helix. Okay, so now each of the double helix gets replicated or duplicated, and the process is called DNA replication. Now, what is exactly in that replication machinery? So it actually, for one DNA strand to get duplicated, like from the parent strand, synthesis of daughter strands, actually requires a good lot of enzymes and certain different factors, different kinds of factors, signaling pathways and stuffs. So what are exactly the requirements in general for a replication machinery to assemble and start DNA replication? So the things are, one is primase enzyme. Again, any kind of proteins which has, whenever this ASE thing is attached at the end of any kind of enzyme, it is it is having a catalytic activity. Okay, so one is primase. So this enzyme synthesizes the primer at the site of replication to start replication. Okay, so enzymes can't really directly come into that particular site, and then it can't really start synthesizing the stuff okay, directly. So it needs to have some landing pad where it can actually sit and start, like it should have a start signal for it to start synthesizing the strands. All right. So there is primase enzyme which actually synthesizes the primer at the site of replication to, you know, uh, send the signal that, okay, we have to start replication here from here. Then comes DNA polymerase. So this enzyme is actually responsible for synthesizing the DNA of pre-existing, like synthesizing DNA from the parent strand. And however, these are incapable of performing de novo DNA synthesis, like de novo as in right from the beginning. So it needs to have some primer onto which, like from which it can eventually add the nucleotides. It can't really add the nucleotides, like it can, so the primase enzyme, without the addition of any primer, it can't really start synthesizing or start adding nucleotides one after the other. All right. So there is this DNA polymerase enzyme, which is incapable to perform DNA synthesis, de novo DNA synthesis, but can be able to synthesize DNA in a pre-existing RNA or DNA primer. So it needs a primer to start synthesizing or adding nucleotides one after the other. Now, DNA polymerase can have actually three different types of enzymatic activities. So one is, so there is this directionality that comes into picture. One is 5' to 3' polymerase activity, where it is actually adding in nucleotides in the direction, in the 5 prime to 3 prime direction. Then you have 5 prime to 3 prime exonuclease, where again in the same direction, it is actually exonucleases, it is chopping off, removing nucleotides from the sequence in this particular direction. And then there's again 3 prime to 5 prime proofreading. Okay, proofreading activities where actually the enzymes actually helps in, like once 5 prime to 3 prime synthesis happens, then sometimes it can be, sometimes some misincorporation of some nucleotide can happen in a particular strand. So what happens in there is actually the polymerase has this activity where it can actually go from the opposite direction, I mean the other end, to actually read and check if any like misincorporated nucleotide is there or not. All right. And then there's gyrase, DNA gyrase, which actually is required to unwind the double-stranded DNA. So in general, if you think of a double helical strand which is coiled onto, like onto itself. So if you think about a rope, right? If you disentangle the rope, what happens is it has the tendency to again wind upon itself, right? So there are certain enzymes, there are certain proteins, SSB proteins, topoisomerases and stuff like that, where it helps to, stop or kind of prevent those strands to reanneal and coil with each other. So one of them is gyrase. This enzyme is actually required to unwind the double-stranded DNA helix. Then there's ligase. Definitely now, one after the other, you have the nucleotide being added. Now you remember, initially it needs a primer. The primer is something else, and then with the primer, it is adding one after the other. The nucleotides are being added, right? So now those have to be joined together. So that joining activity is carried out by DNA ligase. Okay, the DNA fragments are eventually joined by ligase enzyme. Then there are SSB proteins which bind to single-stranded DNA templates. Okay, so what happens is when DNA is unwound, then this SSB protein binds to the single-stranded DNA to avoid rewinding, and it helps to reduce the energy required for unwinding. Okay, all right. Now, what happens in replication is first you have to identify where we have to start the replication, right? So it is actually difficult to start replication of a circular or a very large DNA fragment randomly. So it needs to have specific sites. So how are those sites determined? So there's a particular pattern of nucleotide sequence which exists on DNA to recognize and facilitate replication, to facilitate the initiation of replication. So actually the difficulty here with replication is to first you have to denature the double-stranded DNA, right? So there should be some kind of sequence present with low melting temperature so that it can easily unwind in that particular region, and then eventually the required factors bind to it and start copying, I mean replicating or duplicating the DNA strand. So the task is to actually denature the double-stranded DNA, and it needs to have a certain sequence with low melting temperature so as to facilitate replication. Now, as discussed in earlier lecture, the interaction between adenine and thymine is mediated by two hydrogen bondings, right? So, but whereas the guanosine, guanine and cytosine residues have three hydrogen bondings between them, which is type, right? So, interaction between adenine and thymine is mediated by two hydrogen bonding, whereas guanine and cytosine is mediated by three hydrogen bonding. So, as a result, presence of AT-rich region will facilitate earlier denaturation and assembly of replication machinery, right? And these sequences are mostly present in a region, like in a particular region, which is known as the origin of replication, or in short, it is also written as ORI. Okay. Now, this is the schematic representation. Now, what happens is you have the origin of replication, right? Now, the synthesis happens always in the 5 prime to 3 prime direction, right? So it has to happen in the 5 prime to 3 prime direction. So if we see like 5 prime to 3 prime direction, so the other strand will be from 3 prime to 5 prime direction. Now, what will happen here is So for this, it will start synthesizing from the 5 prime to 3 prime direction. For it, the primer will start from, I mean, the primer will be attached here, and then from this part, it will eventually start synthesizing. Like the polymerase will keep on adding the nucleotides, and eventually the fragment will be elongated, right? However, for the other strand, the problem is, the thing is here, it it won't be starting from, because it always synthesizes in the 5 prime to 3 prime direction. What happens here is it is not getting that directionality on this, on the complementary strand here, so it will start synthesizing from there. Okay. However, short fragments will be synthesized, which are actually called Okazaki fragments, and eventually these fragments will be joined by, first, definitely for each short fragments, you see this thicker part here, which are the primers, onto which then eventually the DNA polymerase adds the nucleotides, and eventually these short fragments are joined. First the primers are removed, definitely, and then the fragments are joined together by ligase. Okay, so this is the overall thing what happens in DNA replication. Now, if we go into it slowly, so the unzipping of DNA at the origin of replication, now you see this is forming a Y-shaped structure. So this Y-shaped structure is called a replication fork. And a short chain of RNA is formed at the 5 prime end, which serves as the RNA primer. Okay, onto which eventually the DNA polymerase will keep on adding the nucleotides during replication. So this is the RNA primer. It is synthesized by primase. One enzyme is there which adds on the RNA primer. Synthesis of RNA primer is essential as DNA polymerase cannot perform de novo DNA synthesis. Okay. Now when we talk about the elongation, like eventually the addition of nucleotides, so DNA polymerase can start the synthesis on the pre-existing RNA primer, and incoming deoxynucleotide triphosphate like ATP, GTP, TTP, and CTP, they get joined by hydrogen bonding to the appropriate nitrogen bases of a single DNA chain according to the base pairing rule A to T, T to A, C to G, and G to C. And the triphosphate, the nucleotide triphosphate that joins to each DNA strand, actually, what happens here is it is a triphosphate, right? So there are 3 phosphate groups attached. So what happens is 2 phosphate groups, the high energy bonds from the coming, breaking those high energy bonds actually sets free pyrophosphate molecules. Pyrophosphates are basically, there are 2 inorganic phosphate groups to pi. The energy released by breaking this particular high energy bond is actually used to drive this elongation step or polymerization step during DNA replication. Okay. So the released deoxynucleotide monophosphate joined to each single DNA chain become linked together to form a new DNA chain. Okay. So I think new DNA chain. And what happens here is, so this one where we see a continuous strand getting synthesized, this is the leading strand because this is a continuously synthesized strand, whereas the other strand is the lagging strand where there are different types of short oligonucleotides. Okay, so which there are Okazaki fragments, then eventually RNA primers will be removed from each of those fragments, and then eventually things will be joined together, right? So all this takes a little more time as compared to the leading strand. So this particular daughter strand, I mean this particular part that gets synthesized is called as the lagging strand. The DNA polymerase can polymerize in the 5 prime to 3 prime direction. Now, when the 2 DNA strands are anti-parallel to each other, new strand must be formed on the older strand, okay, in the opposite direction. So, new strand forming continuously in this part is the leading strand, whereas the short DNA strands are synthesized in the 5 prime to 3 prime direction on this complementary strand. So, these are Okazaki fragments which are eventually joined together to give the lagging strand. The RNA primer is replaced by a deoxyribonucleotide and the gap is joined by the enzyme DNA ligase. Okay. Now, when we talk about when does it stop, when should the replication stop? It can't really go on polymerizing or keep on adding the nucleotides, right? So the replication fork proceed to the end of the DNA or meet the terminal region, which contains multiple copies of TER sequences, like which eventually guide the termination of like where these enzymes can fall off, all the factors bound to it can fall off, and the DNA can eventually recoil and unwind. Okay. Like not really unwind, it can wind on to itself and recoil. And the daughters eventually, I mean, daughter strands get separated, and from the parent DNA, you have two daughter helices. So proofreading and DNA repairs. Now, template nucleotide sequence directs the accurate incorporation of incoming nucleotide, and it actually ensures the accurate DNA replication. But after every round of nucleotide incorporation, DNA polymerase actually runs in the other direction, 3' to 5' direction, in the backward direction to check for the accuracy of nucleotide that has been incorporated. So if any error is detected, it is actually corrected at this particular point. So if we talk about DNA replication to summarize the stuffs, so it is a semi-conservative form because the thing is here you see from the parent strand, one new daughter strand is getting incorporated or synthesized, right? So it has one parent strand, one daughter strand. So eventually, each generation of replication, like after DNA replication, what you have is a hybrid molecule of one parent strand and one daughter strand, right? So this is why it is called a semi-conservative nature of DNA replication. It starts at the origin of replication site. It is synthesized by DNA polymerase in the 5' to 3' directional. It can be unidirectional, how we saw in this straight, like linear fragment. It can be bidirectional as well when in case of circular DNA molecules like plasmids or stuff. What happens is you have uh, like if you have this circle here, suppose this is the origin of replication. So replication bubble will be formed here and DNA can start replicating from this side as well as this side. Okay. I mean from both the ends, okay, of the circle. Definitely we looked at it is a semi-discontinuous form of uh, like process where one leads to the formation of leading strand and a lagging strand, and RNA primers are required because DNA polymerase cannot function de novo. Then it is a three-step event initiation where DNA is prepared for synthesis. Proteins bind to the DNA, double helical interactions are broken. Then you have elongation, which is the synthesis phase. Proteins connect the correct sequences of nucleotides into continuous new strand of DNA. And termination, stopping of DNA synthesis, proteins release the replication complex. So the different kinds of proteins that are involved are helicases. It separates two strands. Primase, which synthesizes the RNA primers that is required. That is the very first thing that is incorporated, and then eventually that polymerase incorporates nucleotides. You have single-stranded binding proteins, which prevents re-annealing of the single-stranded DNA templates. You have DNA polymerase, definitely, which synthesizes the new strand. You have certain tethering proteins which actually stabilizes the polymerase, which latches on to the enzyme and eventually helps it in uh, kind of staying on the DNA strand that it is polymerizing. Okay. And you have finally DNA ligase, which seals the nicks uh, but by forming phosphodiester linkage. Now, DNA replication, this particular thing is actually used in a particular, a very commonly used uh, experiment or commonly used method, which is polymerase chain reaction or PCR, which actually, because from one parent strand, you are every time getting like it's kind of an exponential multiplication, right? 2 to the power n number of copies after n generations, you will be making 2 to the power n number of copies. Now, suppose uh, you have maybe you get a certain amount of DNA which is very less in number. So what you do is you actually do polymerase chain reaction, like the same replication is happening in multiple cycles, and eventually you are amplifying the quantity of that one. Suppose you have three strands of DNA, suppose, and eventually you have millions of copies of the same three strands. Okay, that now you can actually do your subsequent downstream processing or whatever you want to do with the amplified amount of DNA. You have enough amount of DNA to actually perform your assays and maybe deduce certain things and evidences against it. So we will eventually look at what are the applications of PCR. So we'll eventually now we will look at what actually happens in PCR. So first, definitely you have to break the hydrogen bonding between, you have to denature the helices, right? Separate the two strands. So this is initial denaturing, which leads to melting of the double-stranded DNA into single strands. And then what you do is eventually lower the temperatures. Now there are, so because for, you saw like you need to have certain RNA primers, right? Similarly here also, it is not really, not necessarily RNA primers, so those are DNA primers only. But here it needs to have certain primers which are complementary to the individual corresponding template strand, right? So those bind at the specific regions, and then there's polymerase, which is, so in general, what also happens is it is a thermostable DNA polymerase, because the thing is, for denaturation to happen, it is at a very high temperature. Now, at a very high temperature, all these enzymes have to be stable also, right? We kind of read in previous sessions that at a very high temperature, proteins can actually denature or degrade, right? So we don't want that with the polymerase enzyme, right? The enzyme shouldn't be non-functional, it should be functional. Okay, we shouldn't be denaturing or degrading the enzyme here. So that's why what we need is a thermostable DNA polymerase, so that it can withstand that a very high temperature of 95 degrees Celsius. Okay, so that eventually after these primers bind to their specific sites at the complementary position, so then elongation happens via DNA polymerase, which ideally should be thermostable. And then eventually now you can imagine, like, first round of synthesis, you have from one strand you have two strands. Now each of the two strands will eventually form, like, give two copies each, right? So eventually you have 2 to the power n number of copies, so which is an exponential amplification, which allows the accumulation of the specific type of DNA strand that we are trying to produce millions of copies. So these, basically the cycle goes on. These are the steps. In general, denaturation at very high temperatures. Then you have annealing at slightly lower temperatures where these can actually bind, and then eventually you elongate. Okay. Now, if we talk about the development of PCR, there are different events. So 1950, there was the discovery of mechanism of DNA replication by Kornberg. He discovered the first DNA polymerases and different factors like helicases and primers. Then in 1976, we have the thermostable DNA polymerase, Thermus aquaticus, was isolated. So Thermus aquaticus is a bacterium which stays in thermal vents. Okay, so if they are staying in thermal vents, so then they will have all their machineries and enzymes and stuff uh, being produced, because they also need proteins to really, I mean, not only proteins, I mean, they need to have all these replication, transcription, translation machinery going on inside their cells to really let them thrive in that, in such high temperature environments, right? So definitely the DNA polymerase isolated from that, these types of organisms which live in thermal vents will be more stable at such high temperatures, where we want to denature the DNA at 95 degrees Celsius or 98 degrees Celsius, like that. Okay. Then there was a scientist in 1983, Mullis, who synthesized DNA oligoprobes. Oligo means more than few number of nucleotides, a stretch of nucleotides, for sickle cell anemia mutation. Then he did repeated thermal cycling to get a small segment of that particular of a gene. Then Mullis and Tom White tried designing experiments to test PCR on genomic DNA, but the amplified product was not visible in agarose gel. Okay, and then patent was filed eventually when this polymerase was purified, the thermostable polymerase. So initially when they tried amplifying it multiple times, okay, so they weren't able to do that because whatever polymerase enzyme they were using, it wasn't stable in such high at such high temperatures. Okay, so eventually when Taq polymerase was isolated, at that time I think only two enzymes, yes, Taq and VST were known. So Taq was found to be more suitable for PCR because it was more stable at such high temperatures. Eventually the development of the, so the instrument where PCR is carried out is called a thermal cycler. Okay, so when we talk about the primers, It needs to have certain length, and it needs to have a certain melting temperature and annealing temperature, where it shouldn't be very high. Otherwise, it will be too tight a binding, and the primers won't be too tight a binding, and eventually it won't be able to, you know, uh, move off and bind to the other template strands and stuff. And if GC content is also higher, then because GC forms like three hydrogen bondings, so there you don't need to like, it's very strong bonding, right? Comparatively. So there, if the primers again aren't able to detach, and then they won't be able to bind, it will be too tight a binding, and hence eventually it won't be able to detach from the template strand and bind to other strands to amplify or duplicate. And what else? Dimers, yes. There are primers. So the primers should bind to the template strand. It shouldn't bind to itself. If it has complementarity, the reverse, I mean, two primers are there, right? For each of the complementary strand. So those two primers should bind to the complementary strand, not to themselves. Okay. So we should take care of that part also while designing the primers. Now, how to really analyze a PCR reaction? Okay, so once a PCR cycle is completed, the amplified product is loaded on an agarose gel and observed after ethidium bromide staining under UV light source. So ethidium bromide, basically what happens is, if you remember the structure of DNA, it is a double helical structure. So there are bases, right? All these nucleotides, complementary nucleotides stacking over the other, right? So what ethidium bromide does is, the base stacking in between the bases, it can intercalate and then fluoresce. I mean, gives off like gives off signal under UV light. So yeah, so that's how you eventually see, and then you run that particular amplified. This is basically a gene only, right? A sequence of nucleotides. So you actually uh, compare the sequence of nucleotides against known like known sizes of nucleotide sequences, and eventually you understand that, okay, this is, suppose I wanted to amplify a 10 nucleotide or 20 nucleotide, or suppose 100 nucleotide long gene gene sequence or nucleotide sequence. So eventually you will see where it is running against known fragments, and then you can kind of check the PC if your PCR reaction has worked or not. And so the enzyme TAC stands for Thermus aquaticus, T for thermus, and then aquaticus species, which is a microbe found at really hot springs in Yellowstone National Forest. And TAC polymerase is stable at high temperatures, and it actually requires magnesium for its activity. And the optimum temperature for TAC polymerase is 72 degrees Celsius. However, what happens is, in the earlier applications that we looked at, the, the 3' to 5' exonuclease activity, where it can actually proofread and kind of remove the misincorporated bases, right? However, TAC polymerase actually lacks 3' to 5' exonuclease proofreading activity, and it has been found to misincorporate one base in about 10 to the power 4 nucleotide incorporations. So a 400 base pair target will contain an error in 33% of molecules after 20 cycles. Okay. So due to this, there are certain alternatives also. There is like PFU DNA polymerase from another enzyme, another organism, Pyrococcus furiosus. So this particular DNA polymerase has 3' to 5' exonuclease proofreading activity, and error rate is lesser than TAC polymerase. However, more amount of primer is added to avoid primer dimering. And what happens is, many times now we looked at PCR, what happens is, we are adding primers that will bind to the corresponding template strands, right? However, what if we don't know the sequence of the template strand only? So what one can do is, um, we are for the very first time we are trying to purify, uh, trying to kind of amplify a particular gene. Okay, so what also happens is, primers are designed in such a way that it is used against closely related species, used against genetic of that genetic sequence that is present in a closely related species. Okay, there can be different kinds of applications of PCR. Uh, in different kinds of diagnostic tests, criminal investigations, parental testing. food science, tissue culture, everyday laboratory stuffs, uh, different kinds of research laboratories. PCR is a routinely used technique. So in diagnostics, suppose PCR in infectious disease, again, uh, you can detect the presence of pathogens. So a pathogen, what if it is present in a small or low, like it's not really present in such a huge amount that it can be detected. But you have the DNA sequence or genomic sequence of that particular pathogen, right? So you can simply design primers and then try to amplify and make millions of copies, and then just check that if the particular pathogen is present in the body or not. Okay. So actually PCR is actually used in diagnosing many diseases like HIV, hepatitis B and C viruses, chlamydia diseases, Neisseria diseases, and tuberculosis, etc. Like presence of these pathogens. Now, in fact, PCR is a very powerful technology to detect the diseases, like for genetic testing as well as for parental testing. Now, suppose if somebody is prone to cancer, and because they have a BRCA gene, and now if you actually amplify it using PCR, the BRCA gene, it can also be done to actually predict whether the person is predisposed to breast cancer or not. Okay, so these are like kind of diagnostic tests, or uh, where you can use PCR to really uh, kind of for prediction whether a person will develop cancer or not. Now, even very recently, we had COVID-19, right? So you remember like RT-PCR, this was a very this particular technique had become a household name, right? Now, so what actually was happening here is, so we looked at reverse transcription, right? So COVID-19 virus, the genomic, the gene, like the genomic content was RNA, right? So now that RNA was reverse transcribed into DNA, and then the DNA was amplified into multiple copies. Suppose someone had some flu-like symptoms, so the content of the virus was lesser, but still to detect if the person can, develop, so that they start kind of distancing and isolation. So one needed to actually amplify that particular DNA content and then eventually see if, uh, I mean, the person has the SARS-CoV-2 virus or not. So what, remember these swab tests and all those stuff. So you actually obtain specimen, and then you extracted RNA from the specimen, convert it to DNA. This is where the RT from RT-PCR term comes into picture, which is reverse transcription. Eventually, the PCR is performed. You amplify the SARS-CoV-2 virus, viral DNA actually, using specific primers, and then you can detect the presence. And then either the nasopharyngeal swab was taken or saliva was taken, right? Similarly, like from saliva, whichever sample was being taken, eventually RNA was extracted and RNA was reverse transcribed to DNA, and eventually the DNA was amplified to produce millions of copies and then detected to see if this particular SARS-CoV-2 viral, like how much is the load of this SARS-CoV-2 virus inside the body. So this is what RT-PCR principles. So this is the same thing that has been talked about. So now RT-PCR, you can actually do it either in one go, which is called one-step RT-PCR, or via two steps. So in one tube only, you actually add all the primers, reverse transcriptase, enzyme, which will actually convert RNA to DNA, and then DNA polymerase, which will eventually, during amplification, it will keep on adding the nucleotides, and then adequate buffers and specific buffers and stuffs. However, this is a one tube thing, one step RT-PCR. Another is to first synthesize. Acha, one more thing, like when RNA is used as a template to form DNA, the DNA that is formed after reverse transcription is called cDNA or complementary DNA. Okay. So in a two-step RT-PCR, first you synthesize the complementary DNA, cDNA, but like first the reverse transcription takes place, and then against that cDNA you have specific primers. You add the specific primer, and then eventually you amplify via PCR. This is called a two-step RT-PCR. Okay. You can also use PCR for genetic testing, right? Suppose a child is there, if to check who is the mother and father of that particular child, one can again use PCR technology to detect. Definitely, we looked at the diseases associated. Even we can actually see if a certain thing is present on a child, is present in the father or mother, or we can go through the genetic sequence of the father and the mother and also check if the child is predisposed to some kind of disease or not. All right. Definitely criminal investigation. So you know that uh, the forensic team which collects evidences from a criminal, like crime spot, right? So site like crime scene or places like that. So they collect maybe hair samples and stuff like that, can have DNA. So what they basically do is they take the DNA from those samples, isolate the DNA from those samples. Now those are very less in number, right? So they again design primers, amplify that particular DNA that is present in the sample, and then match it from the suspect's DNA. Okay. So a sample of blood, hair root, or tissue left in the crime scene can be used to identify a person using PCR by comparing the DNA of the crime scene with that of the suspect or with the DNA database of earlier convicts, right? So this is how this is also used in very such a useful tool in criminal investigation as well. Now, next we looked at duplication of DNA strands, right? Now, this DNA, how it is really transcribing or how is it synthesizing a messenger RNA, or in general, how RNA is synthesized from DNA. So, if you look at transcription, so again, it needs to have a particular start point from where the transcription can start, RNA synthesis can start. So the first base pair from where transcription starts is called the start site, and the enzyme that does that is called RNA polymerase. So RNA polymerase actually works in a DNA dependent manner where it actually sits on the DNA template. Again, this polymerase also synthesizes in the 5' to 3' direction. So for the RNA polymerase, 5' to 3' direction, this will be so 3' to 5' direction will be the template strand, and 5' to 3' direction will be the synthesis, I mean transcription carried out by RNA polymerase in the 5' to 3' direction. Okay. So the other strand, that is the complementary strand as compared to the template strand, basically now if you remember that it is a complementarity, strands are complementary, right? The nucleotides are complementary to each other. Now, if it has ATGC, suppose it has ATGC, so whatever is the basis, whatever are the bases present on the complementary strand, eventually this is what will get incorporated in the mRNA that is being synthesized, right? So, just in case of thymine, in RNA we have uracil. Okay. So, what is happening here is here this particular 3' to 5' serves as a template for RNA synthesis in the 5' to 3' direction. So, but the template, this is the template strand, but the sequences that get incorporated eventually are complementary to the template strand. So, but it is exactly similar to the this particular strand, which is also called as the coding strand. Okay. So also, if we talk about start site, which is the 0th position, okay, upstream of the start site are towards the 5, if we move towards the 5' end or minus direction, that is sequence before this. Suppose if this is the start site, so all the sequences towards the left side will be upstream to the start site. Okay. Now, if we talk about downstream sequences, all the sequences and downstream sequences are represented as plus. Okay. So if this is the 0th position, the start site, then all the sequences downstream to the start site is actually uh, uh, like now is in the plus direction, not towards the 3' end direction. Okay. Now, DNA is a double-stranded structure. During transcription, only one strand is transcribed. Okay. So that transcribed sequence is identical to the complementary strand of DNA, right? So therefore, this is called the coding or sense strand, and the other one is called the non-coding strand or antisense strand or the template strand. Okay. Now, if we look at the differences between prokaryotes and eukaryotes, the transcription machinery. So, prokaryotes have a polycistronic type of transcription because the genes are polycistronic. That means there are no, so all the coding sequences are attached one after, placed one after the other. However, in case of eukaryotes, there are certain interfering sequences between a long gene encoding for a particular protein, okay, or RNA, or in general encoding for a particular protein or RNA. So it is a monocistronic type of transcription. And in prokaryotes, the transcription actually occurs in cytoplasm because it does prokaryotes do not have a well-defined nucleus, and eukaryotes, the transcription occurs inside the nucleus. And another thing is, in case of, so if you look at the nucleus, there are nuclear pores, right? So if protein synthesis machinery, the ribosomes are actually present in the cytosol of the cell. Okay. Now, but the DNA is present inside the nucleus. Now that information from the nucleotide sequence of DNA to actually encode for certain proteins will have to be carried by something which can come out of the nuclear pores easily, and then interact with the ribosomal the protein synthesis machinery, uh, and then eventually synthesize proteins. Okay, so that thing is done by mRNA or messenger RNA, that is the carrier of the message present in the genetic sequence of the DNA, and eventually that is carried on to uh, the ribosome, carried on via the messenger RNA, and eventually those are read by ribosomes and other adapter molecules, and eventually the amino acids are incorporated. So in prokaryotes, because there are no divisions as such, like compartmentalization as such, so the transcription and translation process is coupled, whereas in eukaryotes, transcription is distinct and translation is distinct, and it occurs at very different compartments. Okay. And in prokaryotes, single type of RNA polymerase is required for synthesizing all the different types of RNA. However, in case of eukaryotes, they have three different types of RNA polymerases, which are again required for synthesizing the different types of RNAs found in eukaryotes. But in prokaryotes, there isn't any specific need for transcription factors. However, since eukaryotes are much more complex organisms, it requires transcription factor for initiation. And in case of prokaryotes, the RNA polymerase in question is made up of 5 subunits. We will look at it eventually. And in case of eukaryotes, RNA polymerases are made by much larger, almost double the number of subunits, like 10 to 15 subunits. So in case of prokaryotes, single RNA polymerase is present, which is responsible for synthesizing all the different types of RNA. And eubacterial RNA polymerase is named as hollow enzyme because it is a complete full enzyme, and it has 5 subunits. Okay, where alpha subunit is there, 2 alpha subunits are there, 2 beta subunits are there, and 1 sigma subunit is there. So alpha leads to the assembly of this. This actually makes up the assembly of the core enzyme RNA polymerase, and then you have beta and beta dash subunits, which actually usually performs all the catalytic activities. And sigma, but before any catalytic function and stuff, there is the sigma factor which actually recognizes the promoter sequence on the uh, DNA, okay, on the template strand, and eventually binds, like lets the RNA polymerase sit on that particular area and eventually start transcription. Okay, so alpha and beta dash form the core enzyme, and hollow enzyme eventually is the core enzyme formed by these 4 subunits plus the sigma factor. Okay. So if we talk about the prokaryotic promoter, the promoter typically consists of 40 base pair region located near the 5' end of the transcription start site. Okay, this is the transcription start site. There is, like, there's a long stretch, 40 nucleotide bases upstream of the start site, represented as minus sign here. Okay, so the promoter region consists of 6 base pair consensus sequence elements. One is the Pribnow box or Tata box, this one, and this minus 35 region. So Pribnow box or Tata box is a 10 base pair long sequence um, upstream of the start point, which is like mainly it is called Tata box because it has this particular sequence of TAs joined one after the other, and minus 35 region, as in 35 nucleotides upstream to the start site. This particular region has the consensus sequence TTGACA. Now, what happens here is the RNA polymerase has to bind to the template DNA and initiate the synthesis. So DNA duplex should be opened so that RNA pol can be approached. I mean, RNA pol can actually bind to the single stranded DNA template, and the efficiency of this initiation is actually inversely proportional to the melting temperature. The more tighter the binding, definitely the more it will be more difficult for the DNA duplex to actually separate into individual strands and RNA polymerase to actually come and bind onto the recognize the required sequences, right? So yes, AT-rich region again because they have two hydrogen bonds. These regions have lower melting temperature as compared to the GC-rich region with three hydrogen bonds. And AT-rich region is actually good for melting of the duplex and easy to create an open promoter complex than GC-rich region. And then the sigma factor of RNA polymerase recognizes the promoter sequence. And then the hollow enzyme, which is the four subunits alpha, alpha, beta, beta dash, plus the sigma factor. It binds and forms a complex now, which is known as a closed complex. Okay. And then eventually the sigma factor is released when the chain reaches nearly up to 10 bases, leaving the core enzyme for further elongation. Okay. Now, a closed complex was formed for transcription to start and elongate. Okay. Now, open complex, when this closed complex is actually converted into the open complex by again melting short region of DNA, which might be 10 nucleotide long. Okay. So, RNA polymerase actually binds at the promoter region and eventually unwinds it and covers it. Okay. From suppose from minus 55 to plus 1, as in 55 nucleotides upstream of the start site, and then start initiation here. One template strand is available for the incoming nucleotide for base pairing and eventually RNA synthesis occurs. So the minus 10 region of the template is essential for recognition. The promoter region is double stranded in a closed complex and single stranded in open complex. So RNA polymerase has actually two binding sites for nucleotide. One is the initiation site and another is the elongation site. Initiation site first binds to the first nucleotides within the open promoter complex at the plus 1 position, from where it will start reading the, uh, as in it will start synthesizing the RNA, which is usually a purine A or G. It means first nucleotide will be ATP or GTP. Elongation site bind with the second incoming nucleotide base pairing at plus 2 positions, and then the two nucleotides are joined together. Then the first base is released from the initiation site and the initiation is complete. Now, when we talk about initiation has happened, mRNA synthesis has started. Next, it has to elongate eventually. So the elongation occurs in the 5' to 3' direction. And again, it is carried out by this transcription bubble, where temporarily the DNA duplex gets separated into single stranded DNA because it has to have a single DNA template strand onto which the RNA polymerase can read the sequence and as in bind to the particular site and eventually synthesize RNA. Now, in bacteria, transcription rate is nearly 40 to 50 nucleotides per second at the physiological temperature, and which is again nearly the same as prokaryotic translation rate, which is 50 amino acids per second. Now, RNA pol actually binds to the promoter and creates a transcription bubble, which we talked about here. Then it moves along with the DNA and RNA chain. Then it moves on the DNA template and the RNA chain grows continuously. Okay, this is the RNA. It is moving okay towards this site, and RNA chain is constantly getting synthesized. So the length of the transcription bubble that we talked about here is approximately 12 to 14 bases long, and the length of this particular DNA RNA hybrid, this region, is nearly 8 to 9 bases long. Okay. Now, as RNA polymerase is moving, the duplex eventually reforms again. And like when it moves to this part, this particular whatever transcription bubble was there, the transient breaking of the hydrogen bonds, it can recoil and eventually transcription bubble forms on the next region. So nucleotides are added covalently to the 3' end of the RNA chain, right? Because 5' to 3' the synthesis occurs. And then beta and gamma phosphates are removed from the incoming nucleotides and OH removed from 3-carbon nucleotide present at the end of the chain. Because all the nucleotides are triphosphates, like attached with 3 phosphate groups. However, monophosphate nucleotide added, like attached with a monophosphate, one phosphate group is actually incorporated eventually, and the two high energy bonds from the two phosphates are broken and eventually used for this incorporation. Now, when we talk about termination, the steps in intrinsic termination are two inverted repeats of GCC, like having very high GC content, which is a tighter bond, are present in the DNA template, which is transcribed. Nearly 6 adenine residues follow the second inverted repeat. Now, inverted repeats are forming a hairpin structure which pause the polymerase. Now, due to the formation of stem loop structure, this RNA polymerase is suppose coming here or synthesizing in this direction. So, the AU bonds get broken down, it leads to termination, and RNA molecules get separated. Okay. Yes. And then, so this was row independent termination based on the intrinsic nucleotide sequence of the RNA. However, there is a factor dependent termination also, which is the factor involved in the termination is row factor. So the termination is called row dependent termination. So this type of termination requires row protein. Row is an ATP dependent helicase that disrupts the DNA RNA hybrids. It is actually essential, which causes transcription termination. And row protein is actually a hexamer. Uh, it exists as a hexamer and performs ATP dependent helicase activity, and its subunit has an like a domain that helps it to bind to RNA, like as an RNA binding domain, and another has ATP hydrolysis domain. So these row protein first bind to sequence which is present at the upstream of the termination site. Then these sites are called rat site. Now these sites are very rich in C residues. So the row factor is followed to RNA polymerase until it can't really catch the RNA polymerase. Okay. So row factor then follows the RNA polymerase by its helicase activity, which is driven by ATP hydrolysis. When RNA polymerase reaches at the termination site, then the row protein actually freezes on that structure of the polymerase. And when row factor collapses with the enzyme, RNA polymerase enzyme, the eventually things fall off and termination takes place. Transcription termination takes place. New RNA chain gets released. And in eukaryotes, what happens is the RNA polymerase is much more complex. So the RNA polymerase in eukaryotes, again, in RNA polymerase of mitochondria and chloroplast are much similar to bacterial organisms. However, all eukaryotic RNA polymerases are actually multi-subunit proteins which contain three different types of RNA, which contain multiple subunits, and three different types of RNA polymerases are used for synthesizing different types of RNA. One is RNA pol1, synthesizes rRNA, ribosomal RNA. RNA pol2 synthesizes mRNA. We are talking about eukaryotes. RNA pol3 synthesizes tRNA. Okay. Now, in eukaryotic promoter, each promoter contains some specific sequence which get recognized by the transcription factor. Eukaryotic promoter has no longer has actually a longer region than the prokaryotic promoter because it contains all the sequences which are important, really important for initiation. It also includes core promoter element at which RNA polymerase can get attached and form an initiation complex. And also for efficient transcription, it requires an upstream promoter element. Like promoter site is there where RNA polymerase can bind. Okay, but like eukaryotes, uh, like there's a like before that promoter region, it here eukaryotes have an added complex upstream promoter element, which are basically GC rich region where certain transcription factors bind, and then it helps in binding of the RNA polymerase, and then the transcription can start. So there are different kinds of transcription factors in eukaryotes. So most of them are called general transcription factors. Okay, like TF2D, like TBP, Tata binding protein. Then there are TAFs, TBP associated factors, which recognize the core promoter. The TBP or Tata binding protein recognizes the core promoter, the Tata box present upstream of the start site, and these TAFs actually recognize core promoter, but the non-Tata regions, which helps in the recognition of this Tata box. Then there's TF2A, which stabilizes this TBP binding protein, I mean Tata box binding protein, and the TAFs binding onto the region. Then comes TF2B, it helps in RNA polymerase 2 recruitment, and TF2F, which is again a transcription factor and helps in start site selection. Finally, TF2F, which was recruited by TF2B, helps in RNA pol binding onto the promoter region. Finally, this E factor comes into picture, which helps in the next recruitment of the next factor and modulates the activity of the next factor as well as the helicase activity as well as ATPase activity. And finally, this TF2H factor, these are all transcription factors we are talking about. These helps in the melting of the promoter region with helicase showing helicase activity, eventually um, forming the bubble and RNA uh, synthesis and elongation. So this is the overall sequence in which all these general transcription factors bind. Okay, so these transcription factors sequentially bind to Tata box DNA to form a pre-initiation complex. At last, when this TF2H binds onto the region, it phosphorylates Pol2 like RNA polymerase 2 to initiate transcription in the presence of ATP. Now, when we talk about the termination in case of eukaryotes, again, for polymerase 1 gene, RNA pol 1 gene, it is a row-dependent termination. Okay, for polymerase 3 gene, it is row-independent termination. And polymerase 2 gene is more complex and it is generally coupled with RNA processing event in which the 3' end of the transcript undergoes cleavage and polyadenylation. A poly A tail is added. So poly A tail like poly A dependent termination, what happens in here is it is basically coupled with RNA maturation process in which the 3' end of a nascent RNA undergoes polyadenylation and cleavage, and these 3' end processing reactions are carried out in 2 steps. So what first happens is transcription of this poly A is followed by breaking of the nascent transcript. Then the upstream product is added, like multiple A residues are added, polyadenylated, and the downstream product is degraded. So basically what is happening here is the 3' end is processed and it starts when the elements which are acting at that particular site itself, these are called cis-acting elements. So there are two terms, cis-acting elements and trans-acting elements. Okay, so cis-acting element is, suppose it is acting, so these are certain factors which bind to the same area where the site of action is. Okay, same region or same sequence where the site of action is. However, trans-acting elements are, they will bind somewhere else, but their site of action is completely different. Okay, so now, when these factors actually bind at the 3' end for 3' end processing, it forms a very complex structure which result in high shearing forces. Consequently, the processing slows down, which disrupts the polymerase 2 and DNA RNA hybrid complex, and eventually transcription termination occurs, right? Now, we were talking about the processing of mRNA. So, mRNA is processed, which is called where a cap is added at the 5' end and a poly A tail is added at the 3' end, and eventually at the cap, methyl group as well as a guanine residue is added, uh, eventually leading to capping and tailing of this particular eukaryotic mRNA, and tailing is carried out by poly A polymerase enzyme. And then another RNA processing step occurs, because in eukaryotes we looked at that it is a monocistronic RNA, right, where there are in between the coding sequences there are interfering regions, so which is the coding sequences which are distributed are called exons, and introns are the interfering sequences in between, right? So those introns are actually removed, and eventually the exons, whichever exonic sequences are there, those can be actually joined in different ways. so as to give rise to different reading frames, different types of nucleotide sequence, which eventually give rise to different types of proteins. Okay, so this particular removal of introns is called splicing. Okay, um, and then there are four classes of introns: group 1 introns and group 2 introns, which are both self-splicing introns and does not involve any protein enzymes. Then there are spliceosomal introns. These are not really self-splicing introns. They need certain complexes, spliceosomal complexes, spliceosomes basically, to help in removal of these interfering sequences. And then there are introns that require only ATP for intron removal. Okay, so here splicing, if we look at the splicing mechanisms of group 1 and group 2 introns, so splicing mechanisms in both these groups involve similar steps where two trans esterification reactions take place, where the 2' ribose or 3' hydroxyl group makes a nucleophilic attack on the phosphorus and a new phosphodiester bond is formed. However, the mechanisms of these groups differ in nucleophile which is used, and group 1 actually uses a 3' hydroxyl group of guanine nucleotide here. Okay, and group 1 introns are found in some nuclear, mitochondrial, and chloroplast that code for ribosomal RNAs, messenger RNAs, and transfer RNAs or tRNAs. Okay, and group 2, again here the adenine residue, OH of the adenine residue attacks here and thereby forming a lariat, and this particular thing is spliced off. Okay, and this is alternate splicing that I was talking about. So here these are the interfering sequences, the green ones. Okay, and when suppose this is joined in this sequence, it leads to encoding certain type of protein. Again, if this particular sequence is gone, then it has this combination of sequences which leads to again a different protein sequence or peptide sequence. Okay, so just as an example, what happens is, so this is called alternative splicing. Okay, now what happens here is, due to alternative splicing, functional genes are produced in females and non-functional genes are produced in males. As an example, so alternative splicing occurs using two mechanisms. One, when two poly A tail or cleavage sites are available in the primary transcript. Okay, so it can also, this alternative splicing event can also help in producing the different variable chains in immunoglobulins. Okay, and again there can be two different, that is what I have been talking about. So it can be used in forming or synthesizing two different hormones. Okay, because the protein sequences eventually will be different because the nucleotide sequences get changed. And when we talk about ribosomal RNA, how is a ribosomal RNA which eventually folds and forms the ribosomes which drives the protein synthesis machinery? So how is a ribosomal RNA processed in bacteria? What happens in ribosomal, the rRNA precursor in bacteria is a 30S ribosomal RNA, which is modified and cleaved to give 23S ribosomal RNA, 16S ribosomal RNA, 5S ribosomal RNA, and some tRNA fragments in between are present. Now, this 30S pre-ribosomal RNA transcript actually consists of 16S rRNA sequence followed by a spacer, which can have certain tRNA fragments in some cases. Then there is this longer 23S rRNA sequence followed by this shorter 5S RNA sequence at the 3' end. Okay. Now at times there are more than one tRNA sequences after the 5S sequence, 5S fragment. However, it actually differs in number and sequence of the tRNA segment. So the number also varies, like whatever sequence is present after the 5S fragment, the number, how many of those sequences leading to tRNA formation are present, and what is this sequence composition, that also, nucleotide composition, that also varies. Okay. Now what happens in here is there are 7 different genes for ribosomal RNA in E. coli. Okay, and they are essentially similar in sequence of the ribosomal RNA segments, but they actually differ with the number and sequence of tRNA segments after this 5S fragment. Now, the maturation process involves methylation of 30S ribosomal RNA precursor at specific sites occurring at the 2' hydroxyl groups of bases, and some bases like uridine is there, which is modified to pseudouridine or dihydrouridine. And further, the cleavage process is carried out by there are different kinds of enzymes, RNase 3, RNase B, RNase E, at these particular sites, and then intermediate products are formed. 17S, tRNA, 25S, and 5S, and then further certain nucleases acted to give the final product of 16S ribosomal RNA, which make up the smaller ribosomal subunit. Then 23S rRNA, which makes up the larger ribosomal subunit. Then you have 5S rRNA, and eventually it gives tRNA fragments as well. And in case of eukaryotes, there's a 45S precursor which is formed by RNA polymerase 1 and processed to give 18S, 28S, and 5.8S ribosomal RNA. And 5S ribosomal RNA is transcribed by polymerase 3 from a separate gene in case of eukaryotes. And okay, so also this particular ribosomal RNA processing in eukaryotes takes place with the help of small nuclear RNAs, which are also called as SNO RNAs. Okay. Now, in case of tRNA, the processing in both eukaryotes and prokaryotes, what happens is there is a long precursor of tRNA. tRNA is usually 77 to 78 nucleotides long, and this is the basic structure. Okay, and if we talk about the structure, it has a clover leaf structure or L-shaped structure. All right, and so this is the first residue, and this is the this is the 5' end, this is the 3' end. Okay, so processing of pre-tRNA involves cutting off the extra sequences of by endonucleases like RNase P at the 5' end, this part, and RNase D at the 3' end, this part. Now, RNase P, which acts at the 5' end, is a ribozyme with RNA exhibiting catalytic activity. And after removing these sequences from the 3' end, the CCA is added via another enzyme which is called nucleotidyl transferase. And this enzyme binds to the CCA sequence at its active site, and phosphodiester bond is formed with the 3' end. Now, there are different bases that are modified in a tRNA like N2-methylguanosine, 4-thiouridine, uridine is actually kind of as in modified to pseudouridine. Then there's acetylcytidine. Okay, these all are incorporated here. You can see in this figure the corresponding bases are written here. Okay, so there are different kinds of modified bases present in a tRNA molecule. Now, when we talk about the genetic code, so mRNA is a random sequence of nucleotides, right? Attached with the nucleotide composition is adenine, uracil, guanine, and cytosine. So these three nucleotides together code for specific amino acids and are called as codons. So why three nucleotides, right? So the genetic code is a triplet code called as codon. It is known that we have only 4 types of nucleotides that make the whole genome. So the permutation combination led to showing that each codon consists of 3 nucleotides, which means there are 4 cube or 64 possible amino acids. However, since there are only 20 amino acids, it is obvious that more than one codon codes for single amino acid. So this also illustrates the Bogle hypothesis. And when we talk about the genetic code properties, it is triplet or codon. Each coding sequence has a start and a stop codon to initiate and terminate protein synthesis. Start codon is usually AUG, which codes for methionine amino acid, and stop codons are UAA, UAG, and UGA. In some cases, starting codons are GUG or UUG, suggesting Bogle base pairing. The code is unambiguous, which again refers to the process where the phenomenon when the code is encoding for only one amino acid. And there is no comma or gap in the code, like it will be continuously read as in group of 3 nucleotides. And the code is degenerate. This means that one amino acid has more than one codon. For example, phenylalanine is specific to two codons, triple U and WC, and only tryptophan and methionine are encoded by a single codon. And again, the codon is non-overlapping. Suppose if this is the sequence, then the codons will be AUG, CUG, GGU like that. It won't be like AUG, then UGC. It won't be like that. It will be strictly three nucleotide triplets. And genetic code is actually universal, which also suggests a genetic code and its meaning is common for all life forms. UGA is a stop, but there are some exceptions as well, where suppose UGA is a stop codon. However, in mycoplasma, it actually encodes for tryptophan. And also in certain mitochondrial protein synthesis, there also UGA is not a stop codon. Okay. Now, if we look at the transfer RNA, this is what we looked at the transfer RNA sequence and stuff. Okay. And there are 30 to 45 different tRNA in prokaryotes and 50 different tRNA types in eukaryotes, which suggests that there is more than one tRNA for single amino acid. Now, when we talked about the different kinds of ribosomal subunits, right? And so in eukaryotic and prokaryotic assemblies, so the 30S and 50S actually combine to form a 70S ribosome. Now, whenever we talk about S, I think we talked about in the very early session where we talked about Swedberg unit or Swedberg coefficient, which actually tells about how sedimentation coefficients work in ultracentrifugation, right? So the smaller subunit is 30S, larger subunit is 50S in bacteria, right? So these values are not really additive. When both the complete assembly occurs, it is not really 80S, it is 70S because these are not really additive, but these sedimentation coefficients actually depend on the shape, size, and density of the particles, and basically depends on refers to the time it takes for it to really settle down after centrifugation, right? So, which eventually is affected by the shape, size, and density of the particles. Okay. When we talk about mechanism of translation, so this actually acts as this is an adapter molecule, tRNA. So this needs to be activated. This has to be joined by based on the triplet that it is reading on the mRNA. It will eventually add the corresponding amino acid at the 3' end. And so what is but for that with this process is called charging of the tRNA. Okay. So during this process, amino acids are attached to the tRNA in the presence of enzyme amino-acyl tRNA synthetase, and this enzyme activates the amino acids by attaching covalently to the tRNA, and the tRNA gets charged. And with that particular, suppose if methionine is getting added to the tRNA, then it will be called as methionine tRNA. Okay. During this process, amino acids are attached to the tRNA with high energy bond. So it is called as activated amino acids. Hmm? So amino acids are there. There is this tRNA, and in the presence of ATP, this enzyme catalyzes this reaction to form amino-acyl tRNA, releases AMP and inorganic phosphate. Now, when we talk about the translation pathway in general, so what happens is during initiation, there is this 30th ribosomal subunits. There are three different factors, IF1, IF2, which is a GTP binding protein, and IF3. There is this mRNA, and in case of bacteria, the tRNA is formyl methionolated. Okay. So, which is in brief called FMET tRNA. So, this is binding here, and this eventually goes here, and all the factors assemble onto the 30th ribosomal subunit. Eventually, when the large ribosomal subunit binds, GTP hydrolysis occurs. It leads to, so as in the binding of 50S takes place only when the complementarity between AUG and the anticodon region of the tRNA is established. This is the anticodon. This actually binds with the start codon of the mRNA. Okay, so this is the anticodon which is binding to the start codon of the mRNA, and then this is what is actually the complementarity eventually helps in deciding which particular amino acid will be attached to the tRNA. So after that complementarity is established, then the larger subunit comes in, GTP hydrolysis occurs, factors are released, and finally the 70th initiation complex forms. IC refers to initiation complex. Now the mRNA in like in case of bacteria, the mRNA is this is the start codon, and few nucleotides upstream to the start codon there is something called as Shine-Dalgarno sequence, okay, which is a purine-rich sequence. So the ribosomal machinery, when it has to read the mRNA code, uh, mRNA codons, so it it doesn't really directly start from here. I mean, in most of the cases, it doesn't start from here. Sometimes it needs a landing pad, okay, or a binding site, which is called a ribosomal binding site, or RBS, where it binds and then eventually it scans through and then uh, comes across the start codon and starts synthesizing the polypeptide. And in case of prokaryotes, the SD sequence is actually complementary to the 16S ribosomal RNA. So in general, the ribosomes are made up of ribosomal RNA and ribosomal proteins. So the ribosomal RNA in case of 30S is 16S ribosomal RNA. And at the 3' end, there is a sequence which is called as anti-Shine-Dalgarno sequence, which is complementary to this purine-rich sequence on the mRNA. So this actually helps in docking of the mRNA onto the ribosome. Okay. And if we talk about the differences between the mechanism of translation in eukaryotes and prokaryotes, in case of translation initiation, uh, there are definitely the eukaryotic initiation is much more complex. However, in eukaryotes, there is only one start codon and it codes for methionine and not N-formyl methionine. And definitely eukaryotic cells needs much more, like more than 10 factors, 10 initiation factors, whereas prokaryotic initiation need requires only 3 initiation factors. So eukaryotes process of association of mRNA with smaller subunit is more complex than prokaryotes. And definitely the mRNA is also processed, right? After transcription, it is 5' methyl, it has a 5' cap, and there is a scanning process involved wherein initiation codon is recognized. This recognition is aided by ATP dependent helicases that hydrolyze ATP. And in case like kind of similar to the Shine-Dalgarno sequence in prokaryotes, there is a Kozak sequence in case of eukaryotes at the 5' end. Okay. And again, in case of elongation, there are elongation factors which brings in the tRNA, helps in, again, these are also GTP binding proteins, which helps in bringing in the tRNA corresponding to the codon on the mRNA and like charged tRNAs. And then eventually GTP hydrolysis occurs, and then the elongation factors are released. Finally, what happens is there is peptidyl transfer. So there are different sites on the ribosome, A site, P site, and E site. So what happens is the very first initiated tRNA, the very first tRNA that gets incorporated at the P site. Okay. And then eventually the rest of the tRNAs get incorporated at the A site, right? Now what happens is, again, the peptide bond gets formed here between the P site and A site amino acids, and hence the eventually one by one tRNA comes in with the respective uh, amino acids, and one by one these get added onto each other and the peptide chain gets elongated. And finally, there is translocation. So three things are necessary. Deacylated tRNA is removed from the P site, peptide tRNA moves from A site to P site, and ribosomes should move on the mRNA one codon down. So next codon can come at the A site. Okay. And finally, termination happens. Again, there are stop codons which does not really code for any amino acid in the general scenario. So there are three stop codons, UAA, UAG, and UGA. And whenever whenever the ribosomes encounter a stop codon, it appears at the A site of the ribosome. It leads to termination because there are no tRNAs present corresponding to these codons, and tRNAs do not bind. However, during termination, there are certain factors which come into picture, which are called release factors. So whenever any stop codon is in the A site out of which UAA or UAG is in the A site. RF1 binds to the chromosome, the ribosome. And when UAA or UGA is in the A site, RF2 binds to the ribosome. RF3 is a type of GTPase whose main function is to catalyze the release process via GTP binding and hydrolysis. So if we talk about release factor 1 or release factor 2, it binds to the ribosome near to the A site. Then polypeptide chain is released from the ribosome via peptidyl transferase complex. Peptidyl transferase complex transfers the carboxy terminal residue of polypeptide chain from tRNA of P site to the bottom molecule. And then the release factors and GTP are released and tRNA is also freed. So now the 70th ribosome is unstable due to the presence of initiation factors IF3 and IF1. And there are certain ribosome recycling factors also. And again, the 70th ribosome is disrupted and dissociated into 50th larger subunit and 30th smaller subunit individually. And again, it is ready for another round of initiation. Now, after translation, after the synthesis of proteins, what we have here is the proteins can get like how we looked at the mRNA and post-transcriptional modification. Here, the proteins can also get modified post-translationally, right? So there are different kinds of groups that it gets modified at different kinds of amino acids. Different amino acids get modified, like it can be phosphorylated to certain amino acids, like phosphate group can be attached to serine, threonine, or tyrosine amino acids. Then it can be glycosylated, where a sugar moiety is attached onto an N or O atom in an amino acid side chain. It can be ubiquitinated, where a ubiquitin residue is added to the lysine residue of a target protein for degradation. Then summoylation happens where a small protein is added to a target protein, and disulfide bond is covalently attached to the S atom of two different cysteine residues. It can be acetylated as well, or a lipid moiety can also be attached. A methyl group can also be attached, and certain residues like lysine or arginine, or uh, hydroxyl group can also be attached to certain side chains of amino acid residues. So what is actually the significance of these post-translational modifications? So proteins are actually synthesized by ribosomes. We looked at it, which may then undergo modifications to form the mature protein product or polypeptide chain or mature protein, right? Now, what happens here is post-translational modifications of proteins, which are not really gene or template based, can actually regulate the protein functions by causing changes in the protein activity, right? It can also eventually dictate where these proteins will localize inside the cell, and even it actually affects the different kinds of interactions that a particular protein can have with the other biomolecules or macromolecules inside the cells. So PTMs or post-translational modifications have like really significant biological functions, which include it helps in proper protein folding. It actually helps in stabilizing the protein molecule. It helps protect, like, it actually protects protein against cleavage. So there are protein, we looked at protein sequencing, right? So there we looked at certain enzymes or proteases having specific recognition sites, right? So if some of the amino acids get modified, get like some kind of residue, some kind of modification is happening on some amino acid residue, then that particular cleavage site won't be recognized by a protease enzyme, right? So the cleavage sites are blocked and proteolytic enzymes can't really recognize, and hence the protein is protected from cleavage. Also, some kind of modification helps in the localization of protein, okay? And also the protein activity and function gets regulated. So if phosphorylation takes place, it leads to uh, the active or inactive forms of a particular protein. Acetylation can also regulate diverse functions like DNA recognition, protein-protein interaction, and stability. Then there are PTMs, which are important components in cell signaling. For example, pro-hormones are there, which are converted to hormones, right? And definitely, it eventually the more modifications are added, it eventually leads to diverse kinds of functions, and it makes the proteome more complex. Okay, so we will look at few of the questions. So mutation occurs in the promoter region of a gene, preventing binding of the RNA polymerase. So which of the following will most likely occur? So if promoter region is where the RNA polymerase is binding and then it is starting the transcription, right? So what will happen is DNA replication stops? No. mRNA synthesis decreases or stops. Protein folding is affected. With protein folding and stuff, mRNA can't remain unchanged because and translation rate increases. So translation, wherever protein folding and stuff is coming into picture, we are talking about mRNA synthesis, right? We haven't still gone into protein synthesis. So these two options anyways goes off. So the correct option will be mRNA synthesis decreases or stops because then it will actually hamper if any mutation is there in the promoter region. It can affect the binding of, it is actually preventing, not really affecting, it is actually preventing the binding of RNA polymerase. So the mRNA synthesis will either stop or it will decrease. Okay, because promoter regions binding of RNA polymerase at the promoter site is actually essential for transcription initiation. Okay. Next, a researcher introduces a synthetic mRNA directly into a cell. So, which step of the central dogma is bypassed? So, if mRNA is already there, a synthetic mRNA is there, then there was no need to synthesize mRNA. That means there was no need to transcribe the mRNA. है ना? So, the correct option will be option B. So, which enzyme is primarily responsible for relieving supercoiling tension ahead of the replication fork during DNA replication? Helicase is basically cleaving, I mean, helps in resolving the or separating the two DNA strands. But to sustain, to prevent those strands from reannealing, there is DNA ligase is a joining enzyme. It joins the bases, and then primase is addition of synthesis of the RNA primers that are required for the start of the DNA replication, right? So topoisomerases are the enzyme which helps in relieving the supercoiling tension. Now, mutation eliminates the 3' to 5' exonuclease proofreading activity of DNA pol 3. So, which outcome is most likely? So, replication won't stop completely because it still has the 5' to 3' polymerizing activity intact. RNA primer synthesis stops. DNA polymerase is not involved in the synthesis of RNA primers. Okazaki fragments cannot form. There is no point in not forming Okazaki fragments because Okazaki fragments are formed from like in a 5' to 3' direction, right? So what happens is fragments are formed, but the proofreading activity, which is in the opposite direction, so that doesn't take place. So in case there is any misincorporation of any nucleotide, uh, while polymerization, then the mutation rate can increase during replication because that activity is gone now. It is not really there in the DNA polymerase. Correct option will be option B. Which of the following would be observed during DNA replication? Which of the following would be observed during DNA replication? DNA unwinding stops, RNA primers cannot form, Okazaki fragments cannot be joined. Oh, the question is okay. I think the question is misformed here, misstated here. So it will be like if there is any uh, mutation in DNA ligase, then what will happen? So if DNA ligase problem is there, DNA unwinding ligase is not involved, helicase is involved, right? And Okazaki RNA primers, RNA primase is involved. Replication fork formation, again, there are different kinds of helicases and SSB and topoisomerases and stuff like that. But ligase comes into question whenever we have to join the DNA fragments. So these DNA fragments, which are called Okazaki fragments, cannot be joined if there is any mutation in the DNA ligase. Okay, so I will maybe I can change now also. Observe during DNA replication. If there's a mutation in DNA like this. Okay, I probably didn't frame the question properly. So, oh, sorry. if DNA pol 3 could synthesize DNA in the 3' to 5' direction, what would be the consequence? The consequence would be, um, the answer is coming off already. Yeah. So if 3' to 5' direction also the DNA pol would be able to synthesize or replicate DNA, then there won't be any lagging strand, right? Because it can synthesize in both the directions. So Okazaki fragments would disappear. Again, a continuous strand will also be synthesized in the opposite strand in case somehow DNA pol 3 has this activity as well. Okay, so the correct option will be option A. There won't be any lagging strand or Okazaki fragments, right? Now, which subunit of RNA poly is responsible for promoter recognition during prokaryotic transcription? Answer will be D, sigma factor. In bacteria, transcription and translation are often coupled. Which feature of the prokaryotic cells allows this? There is no compartmentalization, right? So, absence of a nuclear membrane, right? Which property of the genetic code explains why multiple codons can encode the same amino acid? It is called degeneracy. And which of the following are termination codons or stop codons in the genetic code? It is UAA, UAG, and UGA, which are also called as actually nonsense codons because they do not code for any amino acid. This is the general scenario. There are exceptions, but in general, universally, in most of the cases, they are stop codons. They do not have any corresponding amino acid. Now, a deletion of one nucleotide occurs in the coding region of a gene. Okay, so what is most likely the consequence? So the consequence will be like a nucleotide genetic code is a set of three nucleotides, right? So if there is one mutation, suppose yeah. So if suppose this word CODING, okay. Now there is one mutation, that means D is gone. So the whole reading frame changes, right? It will be COI, then NG and R, right? So the whole reading frame is changing. So this is what is called as frame shift mutation. Okay, so the whole reading frame changes and hence it will lead to frame shift mutation. Now, mutation alters the anticodon loop of a specific tRNA molecule. Anticodon loop is, if this is the mRNA, this is the, suppose, maybe anticodon loop. That is also three bases which anticodon sequence and codon on the mRNA are recognized, complementarity is established, and then translation starts, right? But if that anticodon loop only has a mutation and it gets altered, so what will happen? So what will happen is there will be incorrect pairing between the codon and the tRNA. Okay. Because failure of ribosome assembly, there is no ribosome assembly is anyways happening, right? Uh, Even when the factors are joined. Inhibition of mRNA synthesis, no, because all the individual regions are there. Correct sequence won't be there, but mRNA and also mRNA, we are talking about mRNA synthesis first of all. So here this is in context of protein synthesis. So mRNA is already present. And loss of RNA polymerase activity, again RNA polymerase is used in the context of transcription, right? But we are talking about translation here. An antibiotic specifically blocks the A site of the ribosome during translation. Okay, which process will be inhibited? So if the A site of the ribosome is blocked, then the entry of amino acid tRNA to the ribosome will be uh, blocked at the site, because initiation complex formation won't be blocked because the very first tRNA, the start, which reads the start codon, actually binds at the P site. Okay. Binding of mRNA to the ribosome, no. Release of polypeptide chain and entry of amino acid tRNA into the ribosome. This will be the next, eventually the next tRNAs with the other corresponding amino acids. The entry of those tRNAs will be blocked. Now, mutation inserts three nucleotides within the coding region of an mRNA. What is the most likely consequence? So, what will happen is, earlier we saw that suppose this word coding, D gets mutated or removed altogether, deleted. So the reading frame changes, which was frame shift mutation. However, now what will happen here is, now if you have, suppose you added another THE after THE, right? So the reading frame stays the same. It will read it as a set of three only, THE, then COD, then ING, right? However, what will happen here is uh, eventually the reading frame is not changing, but there will be THE corresponding to it. Suppose amino acid A is getting incorporated, and then another THE, then there will be another amino acid A here, which ideally shouldn't have been when only one THE was there, right? So similarly, the reading frame stays the same. However, there is an incorporation of an extra amino acid here, right? So what is the most likely consequence? You have an insertion of one additional amino acid here. Frame shift mutation, no, because the reading frames are fine in sets of three. Complete termination of translation, no, because there is no mention of any stop codon, the ribosomes encountering any stop codon in the mRNA. Ribosome dissociation, no, because I mean, it can easily, this again, there are no stop codons or there are no other defects or any other factors binding onto the mRNA and ribosome that it will lead to the dissociation, right? So everything is working fine, just that additional sequence again in a set of three has been incorporated, the codon or the triplet, and it will insert an additional amino acid. All right. Correct option will be option B. So that's it for today's session. We will we will meet again actually tomorrow with the seventh session. I think on immunology. We'll stop sharing the screen. Yeah. All right. Thank you for joining. I will end the meeting now.